All right, so let's put that over there for now. All right, where are we going to begin this glorious afternoon delight? That is microbiology. Afternoon delight. He's absolutely correct. There is a take-home portion.
How many points is that?
It is slated to be 15 points. So, essentially what I'm working on is trying to make the exam, not to increase the number of questions on the inside portion, but basically take out an essay question. That's my goal. What's that? Yes, so it won't be, my intention is not to make it any longer than the last one. It's actually to make it a little bit shorter so that what you'll have on the back end is those 15 points of the take home. So basically what that take home is it's going to be the data from what you guys did in lab sit for you on Wednesday. You guys will analyze it tomorrow and just give John the raw numbers. I'll put them into an Excel spreadsheet. I will send out that Excel spreadsheet with a list of 3 questions with it or prompts for you to follow. And then it's going to be due Tuesday by 11:59 p.m. So you've got a full extra day after the exam to finish it up. You can start it as early as this weekend though. And I'm just going to create a Dropbox for it. I suppose the one thing that I'll say is I'm putting a lot of faith in you guys that you'll do this alone. So don't let me down. Anyway, yeah, so that's the take-home portion. It'll involve some graphing, it'll include some calculations, it will include some analysis and interpretation. Basically like what we did with the paper last week and on Monday. Similar premise. I'll talk tomorrow about the setup in terms of what it exactly is, but just so that we're broadly in agreement, the types of questions are similar. So there's a similar number of multiple choice, there's a similar number of fill in the blank type questions, and then a similar number of like short response / essay type questions. And I'll talk more about the format tomorrow in class. Any other questions before we get, or general questions before we get going on anything format wise? If you're looking for a heads up on which objectives, again, I'll cover this tomorrow in class too, but just to let you know, every single one of them is covered on the exam with the exception of the last one on antibiotics. We're not going to get it through tomorrow. It's not fair for me to even ask you about it. That'll become something we talk about later on. So determining, like, talking about, I think that last one has to talk about social practices that lead to antibiotic resistance. That'll become a conversation we do probably in about 3 weeks from now. So that'll get pushed off down the road. Yeah, it's the last one of the antibiotic ones. All right. So, where do folks want to begin? Where are we having the most trouble, or what do we want to start with? What was that?
The well.
The whale.
We should just go over it.
Maybe put the exam up there.
Should we do some practice problems just to get us going and see where we're at? Let's start with something rather simple, but one that hits on one of the first objectives. So it is the first one here that says draw and label, or the third one down. Draw and label the 4 distinct phases of a bacterial growth curve. And then it says to compare the events. How about you just which is where? Toward the end. And then these two, first one? Lag here, log here, right? What's log defined by? What's a description of it? Exponential growth, fastest growth of the organism, all work fine. I want to point out something that we didn't really talk about, but I mean this isn't what you guys observed when we did our growth of our unknowns. Really, what you observed was a single time point at the end here, and we looked at 5 or 6 different conditions. So the optical density that they grew to, or where they reached stationary phase, was dependent on that condition. And so that's why I say this is a generalized curve, but it can differ. For example, one of the old exam questions, and we should look at this one real quick. Let me pull it up. I just don't remember which one it's in, so bear with me as I find it. Yeah, it's this one, 16. So it's an example of growth curves. So it says, in this question it says, you measure the growth of Pseudomonas aeruginosa over time in LB media that has been growing at 22, 33, or 60 degrees Celsius. And then it says, which growth curve best represents the growth of Pseudomonas aeruginosa at 22? Now, one thing that's not here that you need to know, it is a mesophile. So, given what you know about a mesophile, what I want you to do is justify which of these growth curves represents the growth of the organism at 22 degrees Celsius. Okay? And then we'll connect this back to what we did in lab as well. So I'm going to take a minute or two to try and answer that question. A mesophile. What does that mean? Grows best at moderate temperatures. Usually we say like around 37 or something like that, right? And when we say grow best, we haven't really defined so much what that actually means. Not only does it reach the highest growth amount, but what does it mean about its doubling? Its doubling is optimal at 37 degrees as well. Its log phase is quickest at 37 degrees. So C there would represent the growth of this organism at that 37 degree temperature. But that's not answering the question. Question's asking which of these would represent its growth at 22 degrees Celsius. So it leaves us with B or C. Oh, B or A, sorry. B or A. Okay, why not A? A is an inhibitory event. There's no growth. Do mesophiles grow at 22? Yeah, slowly though. Do they grow at 60? No. And that's how you would look at this and interpret it. Okay? So the organism's growth rate here, its maximal growth rate correlates also with its, when we say that it grows best at 37, we're not only implying it reaches the maximal, a certain maximal optical density or concentration, but it also grows the quickest over that period of time too. Okay. So B would be the correct answer here. And to justify this, your justification would be something about how mesophiles grow best at 37. That's probably what we're observing in curve C. They still grow at 22, but just that growth rate is slower. And that's shown by what we observe in B there. So when I say that, one of the things that let me go back to then, how did this correlate to the lab? What you really viewed in lab was, if you had a mesophile, what was the growth after 24 hours? In other words, what was this optical density value? And that's what you mapped, was just what's the maximal concentration that these cells actually grew to? If you can mentally correspond that. Should we do another growth one? This, I did it on my computer, sorry. This one right here. The organism, I don't even want you to look at the multiple choice because you can answer this without doing so. Based on the 3 different curves and then the growth of the tube in the tube there, how would you describe this organism? 1 term for each, or 2 terms. I'll give you a minute.
So can you explain the lag phase as like just the start, like?
Lag is really where the cells, yeah, it's kind of the start, but we describe it typically as the cells are changing gene expression to optimize their ability to grow in whatever environment that they're in.
Okay.
So for example, if they are a non-halophile and there's some salt around, they will start to express genes that allow them to survive and maybe grow slowly in the presence of that salt.
Okay. So then you said the stationary phase is kind of like 2 grow and 1 die.
Yep.
So like,
Yeah, so like the amount of death and life is equal to each other. And usually at that state, the cells are usually like stressed in some way. Nutrients are low, so there's not a lot to support more rapid growth, which is why that rate is equivalent.
what's the difference between
So thermophiles are going to grow more towards like 50, 60 degrees Celsius, maybe a little bit higher than that. Mesophiles would be inhibiting their growth at that temperature. This is more like a psychrotrope.
I followed the voice thread from what we put up.
This is going to be more like a psychrotrope at 15. The mesophiles will be
It should be 15.
25 to somewhere in there. Mesophiles are going to be closer to 30 to 37 is where we'll find their optimal. Might be a little bit higher than that too, but somewhere in that range. Same topic. Spring. Yeah, this one. There you go. All right. Somebody give me an answer for the first one. Based on the results farthest to the left, this is what? Neutrophile. Middle? Halotolerant, I would say so. Middle, or third one? Okay. Last one? Okay. Does this organism have catalase?
Yes.
How do you know? How do you know that it has catalase just from these results? Or how could you at least make a strong assumption that it does?
Because it forms with open respiration.
It grows in the presence of oxygen. And we said that for organisms to survive oxygen, or at least to be able to grow in the presence of it, they have to be able to survive these radical forms of oxygen that can form during metabolism. And so, organisms that can grow in the presence of oxygen must have catalase. Okay? or the other enzymes that contribute to surviving those conditions. Superoxide dismutase, which we really didn't talk much about. Catalase is the big one.
Grow in the presence of oxygen.
Mm-hmm.
So, how do you describe the diglycolate tube?
Obligate aerobe.
Okay, that's what
I didn't say it, they said it. He thought you said.
So the answer would be
Yeah, so if we were looking at the multiple choice, the answer for this one would be A.
A. What's a capnophile?
We didn't cover what a capnophile is, so don't worry about it. But grows in low oxygen, high CO2. So like organisms that live in the gastrointestinal tract, many of them are capnophiles. Let's go ahead one more real quick on this. But based on those results above, what method of these of these four would be best to inhibit the growth of the microbe? Before you say it out loud, think to yourself so that everybody gets a chance to practice with it. Room temperature in Celsius is about 22, 23. So in general, when we're talking about inhibiting the growth of a microbe, what are we, in general, when we're trying to inhibit the growth of a microbe with using a physical method for inhibiting growth, what do we want to do, generally? Perfect. Do the opposite of what's optimal, right? So this one we know is halotolerant. We know that's an obligate aerobe. We know that it's a mesophile. We know it's a neutrophile. Given that, which of these answers is the best one for inhibiting the growth? Yeah, heat step of pasteurization would definitely kill some of it, if not kill all of it, but definitely injure most of those cells in order to slow down the growth over a long period of time. And then with the cold step that follows that heat pasteurization, that would continue to inhibit the growth. Okay, how about one more? Where would you find, in your dorm room or at home, I love asking this question, where would you find a psychrophile? Damn, good job. Good job. Freezer, fridge. Psychrophiles like what conditions? Cold. Cold conditions. 4 degrees up to maybe maximal of 10 degrees Celsius. So we're talking, let's put that in Fahrenheit real quick. Somewhere between 40 to 50, roughly, is about the temperature. Okay. So one of the big takeaway points here then, in terms of talking about growth and growth control, we want to make sure that, one, we're familiar with the terminology. So with the set of terminology that refers to the growth of organisms over a range of temperatures, over a range of salt concentrations, over a range of pH values, and then we also talked about oxygen requirements. We want to make sure that we're familiar with those terms. The second part, as we've just kind of hit on, is how do you inhibit their growth? And again, at least when it comes to physical methods, it's doing the opposite of what's optimal for them. Okay. I guess I'm going to pause here real quick. Any questions on those concepts? Sid.
What are the different heat levels?
So we have psychrophiles, which grow best, usually have an optimal temp at around 4 degrees Celsius or so. Psychrotrophs, which will grow best roughly at like room temperature, maybe a little bit higher than that. 22-ish or so. And I say this roughly because you can refer to 2 different resources and they're going to give you different answers for the same organism. Mesophiles, body temperature, so typically like 30 to 40 degrees with a max around 37. Thermophiles, 60, 70 degrees Celsius, roughly. And then hyperthermophiles, boiling water.
What was thermophiles?
Thermophiles are typically maximal around 50, 60 degrees Celsius, maybe even up to 70 degrees Celsius. When I talk about this, I always think of Pirates of the Caribbean, where they say they're more like guidelines, and that's what these are more like guidelines than hard and fast rules of what an organism is. Matt.
So where would you say that this organism,
So let's look at this one real quick then. This one, we will come back to this. I didn't remember what the answers to this one were. This is more starting to correlate the microbes to where we would find them in the human body. Basically viewing the human body as an environment. Actually, many different environments. So just to kind of like go over this real quick though, what's true about the stomach? Like what's one characteristic? So this is a neutrophile. Would it survive in the stomach? No. Dry skin is a desiccated condition. So some salts usually. Would this organism be able to survive that? Possibly. It's halotolerant. And actually, if I'm not mistaken, I think that one might be... Oh, I remember why that's not the right answer. Because of its halotolerant nature, Staph epidermidis and Staph aureus are both skin microbes. They're halotolerant. Moist skin, 30, 37 degrees Celsius. Lots of water actually would support growth very well. In the mouth, possibly, but we often think of this as an anaerobic environment. And this microbe is obligate aerobe, so it probably wouldn't grow all that well. And then the large intestine, no oxygen whatsoever. So this one would definitely not survive in such a condition. No, we will come back to that. That's going to be something that we cover again later on. Yeah. Yeah. What's the ideal pH for acidophiles? 4, 3, maybe even 2 or 1. There are some that live at a pH of 1. 4, 3, 2, 1. So the other thing, other than just thinking about maximal, also think about range. Acidophile means acid-loving. Will it grow at a pH of 8? No, that's a basic pH. So if you see something that is capable of growing only in acidic pH values, it's an acidophile. Acidotolerant organisms are ones that typically grow well or equally well at a pH of 7, 6, 5. Around 4 it starts to decrease or so. Lactobacillus are a classic example of those. Neutrophiles have a peak where, Danica? 7. And many of you had neutrophiles for your unknowns, and you noticed, like, yeah, the max was around, like, it was able to grow at a pH of 5, 6, 7, 8, and in some cases even 9. That's not unusual for a neutrophile. Okay? And then finally, achillophiles, which are pretty rare, but those will grow only in basic pH values. Achillophiles or acalophiles. Allie. Halophile and acidophile? It's what they love. So an acidophile loves what? Acid, low pH. Halo refers to what? It refers to the salt. Yeah, so the halophiles love salt. They require salt to live. You put them in pure water, they'll die. Some grow at 2 to 5. Some of them require more than 20%. It's a huge range. So they need salt to live. If there's no salt in the media, they will die. They will not grow.
So they'll grow at over 2%.
Some of them will. Go ahead.
How would we tell that apart from a halo?
That's where I wanted to go with this. So how do you tell a halotolerant organism from halophile? Halotolerant, again, tolerant, the term means it's tolerant of a broad range. So I'll pull up the figure that I've got up at the top of this one. Halotolerant grow pretty well over a range of salt concentrations. They will grow in media that has no salt. They will grow equally well in media that has up to maybe 6, 7, 8% salt. This is opposed to non-halophiles that they grow best when no salt is present, and you start to see a continual decline as that concentration increases. Yeah, yes, a tolerant kind of has like a plateau, and that's actually true for a lot of these. Like a acidotolerant organism would kind of have like this plateau of ranges where it grows pretty well over the pH values, and then it would drop off. Now, if you were taking the exam and you go, I don't know if this is halophile or halotolerant, okay, and it's, say, not a multiple choice question. If you make a strong argument, that's what I care about. So if you're like, I don't know if it's halotolerant or halophile, it looks like a halotolerant because it has these basic characteristics, which would suggest it's a halotolerant organism, and it doesn't do, it requires some, or it doesn't, it can grow in the absence of salt, but not really well, but I still think it's a halotolerant because of that. I'll give you the points for it, okay? Sometimes there's not always a right answer on these questions. That's life. Sometimes there's not always a right answer. How do you raise your kids? I don't know. There's never a right answer. Never a right answer on how to raise children. There's only wrong answers. All right. Let me see if there are any other. I want to see if we've hit all the objectives real quick here. I want to point out on the objectives for this particular set of information. So we've talked about the stages of growth. We've talked about classifying. We've talked a little bit about just oxygen requirements, but I want to remind everybody that what that 5.3 objective really refers to is not only where do they grow in a thioglycolate, but what does that tell us about whether they perform aerobic respiration or anaerobic respiration or fermentation. And also, do they have catalase, or do they have enzymes like catalase that can break down those reactive oxygen species? So that's what that one is referring to. Meg?
How do you know if it can do fermentation?
Yeah, that's it. So I guess I wouldn't say necessarily, I mean, you would of course do a phenol red test would be the simple answer. Because naturally you've got those just laying around the room, right? Just phenol red tests all over the place to test for any microbe. In reality, if I were to ask a question on the exam and say, it would be more, could this organism perform fermentation? And it's saying that, and your answer would be yes, it can, or yes, it might be able to, or no, it would definitely would not be able to, based on where it's growing.
So how would you, like, where would it grow best in the thioglycolate?
Yeah. So an organism that performs fermentation, does fermentation occur in the presence or absence of oxygen? Let's take a look at 3. So I'm going to draw 3 different ones up here. for each of these 3 different ones, I want you to tell me first, what is it? Obligate, not obligate, aerobic, anaerobic, etc. I want you to tell me what type of metabolism might it be able to perform, and then does it have catalase or not? Okay. So this basically would kind of lead into a compare and contrast type question. Basically, I want you all to know the stuff that relates to We'll come back to that other stuff later. We're going to actually do molecular bio, so we do Monday on playing. Broad overview of central dogma and then how it relates specifically to bacteria. That's all I want you to know. Do that concept map that you guys did as kind of like a preview of what. All right, so we've got 3 different tubes here. Tube A, tube B, and tube C. Middle one, B? Okay, good. Obligate anaerobe. What could be its forms of metabolism in the respect of what we've talked about this year? Fermentation, anaerobic respiration. Okay? So because we know it only can grow in the absence of oxygen, we know it can't perform aerobic respiration. Oxygen wouldn't even be around for it to perform aerobic respiration. Okay? Catalase present, yes or no?
No.
Definitely not. Definitely not. Good. A versus C. Which one's which? Emily? What could they be? Okay, one could be a facultative anaerobe, and what's the other one likely? One is an aerotolerant anaerobe. Yes. So one is one is the other. There's a slight difference, and I maybe tried to do that a little bit. This one has grown evenly throughout. It doesn't give a damn. This one has grown better at the top. Characteristic of facultative, because that presence of that oxygen usually leads to a higher yield of ATP. The other thing that we didn't state is that because of their aerobic respiration, they produce less toxic byproducts than some of the byproducts of fermentation, for example. Acid, bad for a nutriophile, slows down the growth. What types of metabolism might this organism be able to perform? Aerobic respiration, anaerobic respiration, and fermentation. Does it have catalase?
Yes.
Okay, good. What makes a facultative anaerobe, in terms of like this designation, what makes a facultative anaerobe different from a aerotolerant anaerobe? Yeah, it uses oxygen, grows best where oxygen is. If it uses oxygen, that means our facultative anaerobe can perform aerobic respiration. This one does not. Our aerotolerant anaerobe does not. Go ahead, Zach.
So, if it doesn't use oxygen, then why is there someone?
That's a great question. Good follow-up. Why does it still grow at the top? Yeah.
Should there be like a little gap at the top where there's no growth?
Why?
Is that the honey badger?
That's the honey badger.
So is
Why doesn't it care if oxygen's there or not? It can tolerate it. What allows it to tolerate that oxygen? What allows organisms to survive? Radical oxygen species. Begins with a cata, ends with a lace. Throw it together, catalase. All right. It's got catalase. So Zach, the key difference here between, for example, an obligate anaerobe and an aerotolerant anaerobe, they both could perform fermentation and anaerobic respiration. The difference is those aerotolerant ones have catalase or some of those other enzymes that allow them to grow in the presence of oxygen. So in this case, their ability to grow there it doesn't dictate or doesn't have an effect on the type of metabolism that they would perform. It only has an effect on whether, or in this case, the only ability to grow there is because of the catalase. Yeah. So what would happen then if you threw if you were able to put a catalase gene into an obligate anaerobe? How would it look in its growth? It'd look like an aerotolerant. Yeah. That's the only difference between the two of them is the presence of catalase. There's other things, but that's kind of the general characteristic based on what we've talked about. Kaylin first and then Caitlin.
So aerotolerants would not use oxygen.
Aerotolerants do not use oxygen. No. Caitlin. Yeah, that's a great question. So Caitlin asked, what about our micro aerophiles? Low amount of oxygen. These perform what kind of metabolism? Aerobic respiration. Oxygen's present. Do they have the microaerophiles? Do they have catalase? I think the basic answer is yes, but there's not much of an expression of it. At least that's how I typically classify it. So yes, it's there, but it's nowhere expressed as well, or there's not as much or of those types of proteins. Yeah, because it can't handle very high amounts of oxygen. It can only handle a wee little bit of oxygen. So I would likely to with the bacteria. All right, so that's that third objective. We'll come back to the other ones. Spores. Questions on spores? Basically, the key things to know about spores, they are highly resistant to environmental stress. They are metabolically inactive until they are activated to grow by some sort of, like the right conditions. Kind of like a seed requires water, sunlight, love. Which apparently cannot be listed as an ingredient on granola bars anymore. New FDA ruling. You cannot list love as an ingredient in your food items. What types of organisms form spores? We've said that there's only a certain types that can. Bacillus, yes, but not all bacillus. The other one, gram-positive bacilli, yeah, are the only ones that we know that can form spores. So Clostridium and, gram-positive bacilli. Clostridium and Bacillus, the genus, are the two major ones, that, medically speaking, that are capable of forming spores. We'll come back to that later on, because we'll talk about C. diff in a while. Not today. Down the road. Okay. The other ones are how to measure growth, and then how to assess growth has occurred. Do we need to go over CFU per mil calculations or anything like that right now?
We do one. Will you give us the formula?
No.
So it's CFU over volume.
Yes. So the general equation.
Yeah.
No crutches. We're kicking the crutches away for this one. CFU divided by volume times dilution. Typically, this is measured in milliliters, so the volume should be in milliliters.
So, like, the answer to that would be the
label CFU per milliliter.
Yes, yeah, so that's the concentration. So let's do one quick example problem. So let's say that, ooh, I got a good one. I don't know if it's on here. You incubate Pseudomonas aeruginosa in media containing the quat at varying concentrations for 24 hours at 37 degrees Celsius. Assume the concentration of cells in each condition began with 1,000 CFU per mil. Perform the calculations below just in the 4 mg per mil and the 0 mg per mil down here at the bottom to first calculate the concentration after 24 hours of growth. And then the follow-up question, what's the MIC of this particular quaternary ammonium compound? So, first, calculations. I will do one with you, and then you're on your own. So, 4 milligrams per liter. Our CFU is equal to 150. Our volume is no, that's the 4 is the concentration of the quaternary ammonium compound. So what's the volume that we've plated? 0.5 ml. Dilution? 10 to the 0, which is equal to 1. Yeah. All right. So, in other words, this is 150 divided by 0.5 milliliters. 150 CFU divided by 0.5. Sid. Why didn't I say that again?
What is the dilution on the left?
This is the concentration of the quaternary ammonium compound. So what we're going to do is we're going to compare these values to the original one, Sid, to figure out at what concentration do we find this minimal inhibitory concentration. It's going to be one of these numbers. Okay? So 150 divided by 0.5. Mental math, 300. 300 CFUs per mil. All right. You do the next part. Figure it out for this one. 46 CFU, plated 0.1 mils, 10 to the negative 7th dilution.
Are you gonna do like conversions on the exam where we'll have to like go from?
It's very possible I will. Just remember, okay, easy way to do this. Let's do this one. 10 to the negative 7th. 1, 2, 3, 4, 5, 6, 7.
No, I mean like milliliters. No.
Oh, well maybe I will. I don't know. That's a good idea. Sorry, the exam's already written this time. Yes! Before the weekend! Alright. No, I'm not having you convert between mils and microliters.
So they're good.
We'll save that for the practical.
Oh, good.
Yeah.
So even though it's like 10 to the negative 7, like in high school, you know, like the decimal would be after the 10, so then you would have another 0.
1 times 10 to the negative 7th. Sorry.
Oh, it's 1 times.
1 times 10 to the negative 7th.
So should it really be 10 to the negative 6?
No.
So that means 6 zeros.
But it's still confusing.
Yeah.
I get the 10.
Yeah, so go to behind. Yeah, I get it as to the left.
Oh yes, please bring a calculator. Yeah, you should be bringing a calculator because there is one math problem on it. Aw, crap. Aw, Jiminy Crickets. And if you don't have a calculator, you guys have phones, right?
Yeah.
Most people say don't use a phone on an exam. I hold by that rule, except for if you need to do the calculation. So, if you need to use your phone for one of the calculations on the exam, just let me know that you're gonna use it, so that I'm not like going, ah, damn it, now I gotta yell at them for looking at the answers. You can't find them, I wrote them all myself, but I don't know if it will help. That's why there's so many spelling errors, etc. on these things.
You get joy out of writing.
I do, I love writing them. All right, should we do this? I feel like I've lost everybody in doing this math. 46.1. 1, 2, 3, 4, 5, 6. Hey, want to see another quick trick? So, for those of you that don't want to do it this way, or don't want to use a calculator, here's one more quick trick. 1 times 10, or dividing by 1 times 10 to the negative 7th is the same thing as multiplying by 10 to the 7th. Same exact thing. So, another way you could write this is like that. So it's 460 times 10 to the 7th, or 4.6 times 10 to the 9th, or 4,600,000 cells, 600 million cells. Okay? Follow-up question, what's the MIC? Huh? You can tell the MIC because I gave you the starting concentration, didn't I? What's the starting concentration of cells? 1,000. What's MIC stand for? Minimal inhibitory concentration. What's the MIC? Where there are 1,000 cells after, right?
Yeah.
Which of those is equivalent to 1,000 cells?
Oh boy. Two. You're right.
1 times 10 to the 3rd. 1, 2, 3. All right. So the MIC has the same concentration afterwards as it did beforehand. Theoretically, if it's off by a couple, it's not a big deal. Dan,
I don't get why it's 1,000 in the beginning, just because it's like the starting.
yeah, that's the starting concentration. And we said that the minimal inhibitory concentration is the minimal concentration needed to inhibit growth. Fully. Inhibit growth fully. If you start with 1,000 and you end with 1,000, has growth occurred? No. That's why it's the MIC. Huh? Yeah, the 2. If it's 0, it's actually the MBC, the minimal bactericidal concentration. But when we're talking about mix, we're only talking about growth inhibitory. It's not implying that it's killing the cells. So like when we do that, when you guys have seen, like the problem we'll go over again tomorrow where we calculate the MIC, when you inoculate those experiments, you can't see any cells in there to begin with. You see no signs of growth. You give them 24 hours and then they grow up. Same way that you guys did with your experiments for the salt or something like that. Okay. I'm going to pause real quick. Questions? Okay. So when we talk about chemical methods for inhibiting growth, what I want you to know is not every specific one we talked about in class. That would be a waste of your time. It is a huge waste of time. Some people are going, microbiology is a waste of time. All right. What do I want you to know from this? I suppose maybe the best way to describe it is to describe a couple of things. One, I'd like you to have an idea of when would we use these is probably the most practical thing we can do. So for example, if we were to say if we were to say, for example, we're going to use chlorine, would we use it as a disinfectant, a sterilant, or an antiseptic? Okay? And in the case of chlorine, chlorine is bleach. All right? How do we use bleach in everyday applications? To kill bacteria. Do we use it on our own skin? Do we shower in bleach? No. Does that make it an antiseptic? No. It is therefore not an antiseptic. Okay?
Wait, so the difference between an antiseptic and a disinfectant is that an antiseptic is used on skin?
Yes. So it's safe to use on a human being.
But it's to kill bacteria.
Yep, both of them, the goal is to kill bacteria. All right. How do we use bleach then? Do we use it on a solid surface?
Yes.
It's not an antiseptic.
So it is
It's a disinfectant. We use it on surfaces. We use it on solid surfaces. Okay? Inanimate objects. So a sterilant, or something that performs sterilization, when we say sterilization, what do we mean? Kills what? Everything. Chlorine, if left a long enough period of time, yes, could kill everything, but we don't do that with it. Typically, the best things for sterilization, heat. Heat is a really good one. Yeah, the autoclave is an example. And I'm trying to think of things that are practical as well. Emma?
So should we know what phenols are?
No. No.
So, wait.
I mean,
you guys, this is in class, but like on that chart that you had us fill out, and your key online, do you have in there like what you want us to know on the key?
For these types of chemicals,
Like for this?
I do for the antibiotics, yes. But I did not do anything of the sort for chemicals because, to be completely honest, I don't really care all that much. Each year I'm going through the chemicals, I'm wondering more and more why I'm covering them in some ways. And it's important, right? It's important to understand how we can inhibit microbial growth. But I think one of the things that I'm trying to point out here is that it is going to be a huge waste of your time if you go through every single one and go, okay, phenols are disinfectants, and iodine can be used as an antiseptic, and soaps and quaternary ammonium compounds, they are degerming agents. This should be intuitive. If you have lived for the past 19 years, you know that soap washes things off of the human body or off of surfaces, right? That is the purpose. Even dirty words out of children's mouths, it is still used for that. If it removes something from a surface without killing it, what is it? Is it an antiseptic, a disinfectant, a sterilant, or a degerming agent? It's a degerming agent. That's how I want you to be able to use this. So if I say you have a chemical you're using to, to decrease the amount of bacteria on, to actually kill bacteria on skin. So like you're prepping somebody with iodine before surgery. In this case, the iodine is being used as an antiseptic. Okay? Or I might say you are brushing your teeth that also contains 0.3% triclosan in there. The most likely use of this particular combination, what is toothpaste? Could be an antiseptic. It's cleaning, meaning it's what? It's degerming. It's mechanically removing. Basically, yes.
So what did you say it didn't kill it?
No. No.
Did I hear you say that degerming doesn't kill it?
Degerming does not necessarily kill microbes. Degerming will just remove them.
Okay.
Even if there's triclosan in it, it's still just determined.
Depends on the contact time, right? I don't know. I would assume that it's just degerming.
Septic, aseptic. So it's asepsis means there's not.
Yeah, so that was a question that I got earlier today too. What does asepsis mean? We didn't really go over that term. Basically, aseptic technique is the same thing as saying sterile technique. So when you say that something is aseptic, or when you say the term asepsis, it means no bacteria. Don't confuse that though with antiseptic. Okay, don't confuse it with antiseptic.
So does antiseptic necessarily kill, or does it just remove?
Antiseptics kill.
Is it antiseptic and antisepsis different?
So, it's asepsis.
On the 6 different occurrences to make it antisepsis.
Let me check that real quick. Antisepsis and antiseptic is the same, yes. Antisepsis would be the actual process of using an antiseptic. Sorry, thank you for asking that. I know. It's like learning Spanish all over again. In lab today, I practiced aseptic technique by heating my inoculating loop to kill all bacteria off the inoculating loop. No, you could say it's an aseptic process. Again, this is why I avoided using the term asepsis. Antiseptics are chemicals that reduce bacterial number. Okay? Asepsis, when we use the term like, when A is a prefix, it means nada, nothing, void, zero, zilch. Kind of like the U.S.'s chances of making it to the World Cup now. All of you soccer fans out there, really sore subject. But asepsis means void of bacterial life. So when this term gets used primarily is in hospital settings, but it should not be confused with antisepsis or antiseptic.
Antisepsis means what? Is an antiseptic.
Is an antiseptic, yes.
So is it even like worth knowing what asepsis?
No!
Okay.
That's why I didn't cover it. No, absolutely not. Use the term sterile. Use the term sterile technique.
Crossing it out.
Now, you may hear something different from your nursing professors down the road. But tell them that I said asepsis is a bunch of hogwash. Hogwash would be a degerming agent. Actually, probably not. No, really not, would it? All right.
Can you ask like an example question using those again?
Sure. Let me go to the review questions, because I think that's where we'll find the best.
You know how you said that chlorine is not an antiseptic?
Yeah,
Why is it chlorine?
I knew somebody was going to ask that.
You don't have an answer, I don't expect.
I guess the one thing that I would say is my kids have horrible reactions to swimming in pools, so like the chlorine there still does have an effect, but right, it's based on time of exposure. My kids swim for 4 hours at a period of time.
That's nice.
I know, right? Would love to be a kid all over again. Okay.
No.
Need to go to bed. Didn't you? I think she just said thank you. Not going crazy. Baby, still a little.
Well, heck. I'm realizing that none of the questions I have in here relate to it. I suppose one example would be things that I've already brought up. So an example question would be, I guess I've already given some. I think, here's one. So if you've ever had a shot at a doctor before, a vaccine per se, beforehand they typically will, the nurse or the doctor or whomever will take a gauze pad that has been soaked in isopropanol and rubs it on the skin and then tosses it away. Okay? And really the process here is just, it's not to kill the bacteria, it's just to wash that area. In this case or in this context, how is that alcohol being used? It's a degerming agent. That would be an example
So
question.
degerming can go on skin too,
Yes, absolutely.
but like sterilants and disinfectants can't.
No, and really the reason why we don't use disinfectants on skin is because they are harmful to us as well as microbial life. Yeah. I've never thrown a human in an autoclave and probably never will. Right? It's probably a good choice. A turkey on the other.
Because you're washing the bacteria,
Yeah.
not killing it.
Yes. Because the contact time is too short for it to work as a true antiseptic. Oh, I thought that was a hand raise. Sorry, I was using my old school marching band peripheral vision. I thought I saw a hand up over here. I was gonna step my way over. Anyway. Big thing to take away from this particular section on controlling growth. I think we focus a lot more on the physical than we had the chemical. That's still true, and so pay attention to that. But also, the ways that we test for the effectiveness of a chemical are the same exact ways that we test for effectiveness for antibiotics as well. I mean, in all honesty, an antibiotic is a chemical. It's a chemical way to control growth, if you think about it. So, pay attention to techniques like performing broth dilution assays or performing Kirby-Bauer assays or disc diffusion assays as ways to determine the effectiveness of a chemical. One question I could ask, and this is because at least with some of these chemicals, we don't have defined or standardized zones of inhibition like we do for, gosh, this thing is loud. All right, zones of inhibition, we don't have defined zones of inhibition that determine if an organism is sensitive or resistant to most chemicals. We do for antibiotics, we don't for chemicals. Other thing to point out here, microorganisms typically don't gain resistance to chemical controls of inhibition, whereas they do gain resistance to those antibiotics. So there are a couple of ways in which we can analyze those results for antibiotics. One is just testing 4 or 5 different chemicals and determining which works best against the microorganism. The other way we can perform the experiment is test the same chemical on 2 or 3 different microbes and determine on which of the microbes will the chemical work best. Those are typically the analyses that we'll do on these Kirby-Bauer assays. So let's imagine that we have an example where we have yes, it is the same thing. The Kirby-Bauer is the E-test is another disc diffusion type assay, but doesn't get the glorious name of Kirby-Bauer. All right, so if we have 4 discs here, A, B, C, and D, which of these 4 would you least likely use to clean up this microorganism if you were looking for a good disinfectant? Why? Smallest zone of inhibition, which means what? What does that mean about the microorganism? Is it more resistant or more sensitive to that particular chemical than in relationship to the others? Yeah, it's more resistant. Okay, which would be the best option? C. Okay, good. If you can analyze that, you know how to determine effectiveness of chemicals. All right. No. No. Maybe. Instead, filtration. What the devil? Oh. Instead, filtration is going to result in basically just blocking the flow of a microorganism. So, sorry, I'm trying to get this up on my computer real quick too. Okay. So like the other methodologies, filtration can be a method of sterilization. It can be a method of sterilization. In the context of how it does so, what has to be true for filtration to be a method of sterilization? Well, one, it has to make whatever liquid is coming through, has to end up preventing the flow of microorganisms. If it prevents the flow of microorganisms, what has to be true about the pore size? It has to be smaller than the microbe. Good. So typically we use 0.45. We might even use something a little bit smaller like a 0.22 micrometer filter. Both of those will inhibit the flow of bacteria. Filters do not necessarily, though, stop the flow of viruses because they are typically smaller than bacteria, or bacterial parts like LPS. And in medical terms, where this is really important is that we can engineer some bacteria to make, for example, antibiotics. We can engineer them to encode the genes to make antibiotics. We can also engineer them to do things like make insulin. And in those cases, we have to somehow purify that chemical. Well, if you're purifying insulin, if you heat it, insulin's a protein. What happens to a protein when you heat it? It denatures. Therefore, insulin no longer functions like insulin. And so the way to sterilize it would be to filter it. Downside of this is that you can get, if those cells aren't properly treated, you can get LPS. And one of the things we'll talk about later is how LPS actually is a pretty bad thing for us when it becomes detached from bacterial cells. It's an endotoxin. High fever, chills, etc. often ensue when exposed to endotoxin. Does not work in sterilizing solids, obviously. A solid cannot fit through the pore. Okay. Would this work with air? Yeah, and we find it quite commonly. So HEPA filters, or HEPA filters, however you pronounce it. What we would find in hospital settings, those are commonly used as a way so that when air is being recirculated, it's not allowing for the flow of bacteria, fungus, and viruses through those systems. Okay. So that's the basic premise of filtration. Like sterilization, what you get out in the end should be void of microbial life. Unlike sterilization, unlike autoclaving, doesn't necessarily kill the microorganism per se. That was one. What else was the other one? Pasteurization. Thanks, Emma. All right, so pasteurization, 3 basic methods. Old school method, 60 degrees Celsius, half hour. Newer school methods, 72 for 15 seconds, 140 for 3 seconds. Nope. What's common among all of them? Extreme cold, extreme heat, and time of exposure. Again, what's that? Yeah, it's quick. One of the things that we've often talked about is, or one of the things that we've said, is that two things determine Two things determine how well chemical and physical methods work to control or to kill microorganisms. One is the amount or the degree or the magnitude of that pressure, and the second factor is how long. So you notice with the pasteurization here, as the amount of heat increases, it takes less time to result in pasteurization, in inhibiting the growth of those pathogenic microorganisms. If we were to characterize pasteurization as a sterilization, a disinfection, a antiseptic technique, or a degerming technique, how would we classify it? Okay, disinfection, why? That's part of it. The heat and the wetness do have something to do with it. Think about it in terms of the end results, Kaylin. What's different between sterilization and disinfection? Good. Does milk still spoil if it left out and opened in the fridge? Yeah. So it hasn't killed all microbial life. And this would be an inanimate thing. This is not a human being or an animal or anything like that. So that is why this is not an antiseptic technique per se. Okay. What would this not work against? Spores definitely can be resistant to this. The heat for 15 seconds is not enough to kill a spore. Yeah, hyperthermophiles and thermophiles. If we're talking medical importance, though, it really is the spores that we would be most worried about. Yeah. So another example of where this kind of holds is like if you, I think I brought up this example at least one of the two class sections, home canning. If you or somebody you know is somebody who cans vegetables at the end of the season, you don't necessarily pasteurize, but oftentimes you heat for a long period of time, heat those or stew those tomatoes in the jar itself. Why do we do that? Why take the time to do that? Meg?
Just to get all the extras and that little divot within the top.
Ah, that's true. It is to get rid of the oxygen and everything. But why is that important? I mean, think of it kills the bacteria on the tomatoes. Most importantly, kills Clostridium, kills spores of Clostridium. So there are these examples. If you want something to look at this evening on the internet, because who doesn't? There are these pictures from the 40s, 50s, etc., even in some countries these days, of aluminum cans that have bulges or that have completely exploded. The reason why they explode is because the technique to sterilize or to kill all the microbes in that canned food didn't work out properly. And if you have a food that has a soil-borne organism on it like Clostridium that can also sporulate, it survives the high heat. As that temperature cools, you've got nutrient-rich food for the microorganism. It begins to divide, it begins to grow. And Clostridium, they ferment, and they produce a lot of gas in their fermentation. That pressure that gets built up, that gas that gets created, that's what causes those cans to explode. Okay? So there have been people who've gotten sick from home canning because their pressure, when they pressure cook those cans, it doesn't get up to the proper temperature to kill the sporulating bacteria as well as the ones that are just vegetatively growing as well. I got it sidetracked though. Let's get back to pasteurization real quick. But there's a relationship here. So the hot heat and then the very quick chill is going to be enough to kill many microorganisms. Again, from a food perspective, the reason why we do this instead of autoclaving or sterilizing the milk is because it changes the flavor a lot. And one way to kind of test this out is, does anybody live on a farm? Devin, when we talk about pasteurization, it's the purpose behind it is it's enough to slow the growth, but it's not enough of a temperature change to lead to, for example, a change in the flavor or the taste. Another example, my kids now, or these days, they have basically juice boxes of chocolate milk that can sit in just the normal aisle, do not have to be refrigerated at all. Those are ones that undergo this ultra high temperature pasteurization, 140 degrees Celsius for 3 seconds, and it gives it a much extended shelf life over 72 degrees Celsius for 15 seconds. So there's a difference. Also, if you drink coffee like I do, copious amounts, and ever have half and half pasteurized versus ultra pasteurized, half and half, there's a difference in the flavor. I have a friend who never drinks ultra pasteurized because he says it's too sweet. It's caramelized the lactose that's in there. So that's why we perform this, or that's why this process is performed, as a way to reduce microbial growth, extend the shelf life of some of these food items, reduce the pathogens at the same time.
Is radiation one of them too?
Radiation is another one, and that was another one that Emma wanted to go through. So radiation, 2 main types, right? Ionizing and UV. Both of them, in general terms, both of them have the same effect. The goal of radiation is to what? It inhibit growth, but how? How does radiation inhibit growth? Destroys the DNA. Yeah, so, and the difference becomes, are you using this on a surface versus are you trying to get a penetrating radiation? UV works really great on surfaces. Ionizing we need to use to get inside of something. So where this might pertain to daily life, I actually showed this picture, you saw this, Riley, the picture I showed you earlier today. Let's see if I can find my phone real quick and put this up. Or at least find it on the computer. Okay. So I got a picture like 2-3 weeks ago from Jonah Well, for anybody who knows Jonah. This is being used in Madison at some of the local hospitals. This is known as a True D. It is essentially a huge column, like 4 feet high or so. All of these little lights on here are UV lights. The goal behind this is it removes any need for chemical disinfection of a room after a patient has left the bedroom or left the hospital room. So basically this thing is powered or it can be controlled by an iPad. You walk into the room with it, you set it in the middle of the room, you walk out, you press a button, UV lights the room for 10 minutes or so. And that's it. And it's the design is to damage the DNA of anything in that room enough so that it no longer can grow. Okay? Huh?
It works.
Yeah, apparently it works.
And spores?
Spores would be an exception because spores are more resistant. I don't know if the time is long enough or if the exposure, the intensity is long enough. Madison Metropolitan Sewerage District. They use UV light as well to treat their water that's going through and being processed, essentially. So what they have is basically the length that the water travels is from here to the door, and as that water is moving along, it's passing UV lights like this on both sides of where that water is flowing. Again, the time is long enough to kill any of the microbes that will become in contact with that UV light. Okay. The upside of this, becomes cheap in the long run. Also, you don't have to worry about janitorial staff or hospital staff possibly being exposed to pathogens. You basically walk in, turn this thing on, and let it run. Kaylin.
After it's done running for the 2 minutes, when you still have to go and like wipe everything down?
Technically, no. I'm sure that they do, but I don't know for certain. I don't know anybody who's actually used one of these yet. I'd love to talk to somebody who has, though. It's a good question. Again, though, this only works on surfaces, right? This only works on things that come into direct light, into direct contact with the UV light. So if you're talking about, like, imagine you have a porous bed or something like that, this is not going to work to sterilize the bed. Instead, you would need some sort of ionizing radiation to sterilize the bed itself or the mattress. The importance of ionizing, probably the biggest one, or where we're seeing it nowadays, is again treating food. Because there are microbes, bless you, that not only can grow on the surface of some vegetables, but they actually can grow inside some of the vegetables as well. And then let's think of maybe a couple of examples. Where would ionizing radiation be a good thing in the food industry? Tomatoes, yeah. Where else? Think outside vegetables. Chicken, absolutely. Ground beef, yeah. If you want to eat it raw, you gotta ionize the heck out of it. I will admit though, I think the reason why it hasn't taken hold, tell somebody you're going to radiate their food. Doesn't sound like a good thing. But what folks typically don't understand is that this is a short exposure thing, and as soon as that radiation gets flipped off, there's no more radiation. It's not like these pieces of meat are glowing afterwards. I don't think there's enough scientific sense in the U.S. to understand that. So damage is DNA, bottom line.
Then desiccation.
Desiccation. So in terms of desiccation, what do we really mean here? If we're saying something has been desiccated, or if we're using this as a method of inhibition, what are we talking about? Removal of what? Sorry, you said that, but I didn't hear. I apologize. So water removal, or another way to think of it is increase the osmolarity. So a couple of ways that this can work. If you are somebody, or you know somebody who hunts and then makes like venison jerky at the end of November, December, or if you like to make your own dried fruits at home, or if you've ever had an apricot or a raisin or a prune, which is just a dried plum, all of those use desiccation as a method. We remove the water, and by removing the water, the solutes don't get removed. So at the same time as water is being removed, you're increasing the osmolarity of the solution, or that matrix itself. So peanut butter is a great example. There's water in there, not a lot, but there's some. Jams are another good example. It's water-based, but what is helping to, for example, pull water out from the bacterial cells? What do we add to jams if you make these at home? Gelatin and what else? Sugar, and lots of it. Sugar is great at creating a desiccated condition because the high osmolarity of the amount of sugar that we put in there draws the water out of the bacterial cells, dries them out, and prevents them from performing metabolic activity. That water is also important to stabilizing proteins, stabilizing the structure of the DNA. And as that water gets pulled out, they then lose the ability to survive in that condition as well.
That's why they can last so long.
Yeah, oh yeah, that's why these things have incredibly lengthy shelf lives. So like peanut butter, I don't know what the shelf life of peanut butter is. Couple of years, I don't know. Twinkies, incredibly long shelf life. Nothing wants to grow on there. A lot of available water in it. So this is not something that we've covered, but basically when we look at food items, one of the ways to measure how much water is there is to measure what we call the AW, or the water availability. Peanut butter is at around like 0.5. Pure water is 1. Jams are around like 0.7 or something like that. So the reason why, one, when we make peanut butter, we don't add water to it. So the only water that's there is the water that's in the peanuts to begin with. You've also got a lot of oils, which helps to make it a liquid. And so because of the proteins, the sugars that are in peanut butter, the salt that you add to it, those are factors that help to pull water out of potentially any living organism and keep it in the food itself rather than in the microbes. Does that help some? Okay.
So how does the bacteria then not grow, like, though,
Even though there's food around for them to eat?
Yeah, it's like peanut butter's the food, like, so how are they like not like around, especially if they have like long shelf life?
Yeah. Think of it this way. We talked about halophiles. We said halophiles, for example, can grow in high concentrations of salt. There is a relationship between an organism's salt tolerance and its ability to grow at lower and lower concentrations of water. So for an organism that is a non-halophile, they will not grow in peanut butter. They may survive in it, but they will definitely not grow. A extreme halophile, one that can grow in high concentrations of salt, will be able to not only survive, but may even be able to grow in the peanut butter. So they require less water around to divide and grow and live, etc. I hope that kind of helps to answer the question. Okay. Next.
No, anything about radical oxidation?
No, other than that catalase is required when organisms grow in the presence of oxygen to protect them from it. That's chemistry. We'll hold off on that. See you later. Have a good evening. Should we take a break so that people who want to can shuffle out of here? I'll give a few minutes for people to shuffle in, and then we can kind of restart the conversation on growth and stuff like that. Is that okay with everybody? Just take a few minutes to break? Okay.